Hi everyone, this is Miss Moriarty and I'm here to discuss with us concept number 4, which is all about chemical reactions and enzymes. Make sure as you watch this video that you are using your Chem of Life notes and jotting down notes as we go. And so here, a first part of this concept is understanding what is a chemical reaction. That is a process that's going to change or transform one set of compounds into another. And so this involves changing the chemical bonds that join the atoms in that compound. But remember, when this occurs, both the mass and energy involved in the reaction are conserved, meaning that the mass and the energy cannot be created, it cannot be destroyed. The chemicals or elements that engage in a chemical reaction we call reactants, while the chemical or elements produced by the reaction are known as products. Now, in these reactions, energy is released or absorbed depending upon the types of chemical bonds formed or broken. Some chemical reactions simply just release energy, while others will absorb it. Chemical reactions that release energy usually occur on their own and are spontaneous, while chemical reactions that absorb energy tend to require a source of energy in order to absorb. And so, what would be an example of an energy releasing reaction? This would actually be the burning of hydrogen gas or fire. Here we see hydrogen reacts with oxygen to produce water vapor. The energy as a result is released in the form of heat, and sometimes when hydrogen gas explodes, can even be released as light or even sound. While a classic absorbing energy example would actually be photosynthesis that we know takes place in plants. So organisms such as algae or plants we know harness solar energy. And so this is the type of energy that they're going to absorb. The bonds found in the carbon dioxide and the H2O found in our reactants are going to then break and reform to then form our products, which are known as our sugars, carbohydrates, or starches, and oxygen gas. Now, chemical reactions that release energy do not always occur spontaneously, especially absorbing reactions. The energy that's needed for a reaction to get started, we tend to call the activation energy. And regardless of whether the overall reaction releases or absorbs, there's always activation energy involved. And so here is what two our different reactions would look like based on their graphs. So here on the left-hand side, this is an energy absorbing reaction. So as you can see, the reactants, right, or the elements or compounds that are doing the reacting in the experiment tend to be very low in energy, while the products are really high. And so as a result, in an energy absorbing reaction, activation energy is really high. But let's look at the opposite, right? At an energy releasing reaction, our reactants actually are already high in energy. When they break, energy is released, and our products are then much lower in energy. And as a result, an activation energy in an energy releasing reaction is not as high. Now, some chemical reactions that are essential to life would actually happen so slowly in our bodies because they have such a high activation energy that they could never take place on their own. And so within our cells, we carry substances known as catalysts. And these catalysts speed up the rate of chemical reactions without being consumed by the reaction. And a catalyst or enzyme works by lowering the activation energy of the reaction. Now, a little bit about our enzymes. Now, enzymes we call biological catalysts, and that's because they are catalysts found in living organisms. Most of the enzymes found in our bodies actually fall under the category of proteins. The role that enzymes play within our bodies is to speed up then chemical reactions that take place within our cells. Enzymes are very specific in their shape, and so they generally only catalyze or speed up one chemical reaction. Because of that, enzymes' names usually derive from the reaction that it catalyzes. So, for example, the enzyme known as lactase is going to help break down the sugar that we find in milk called lactose. Another example would be sucrase. The enzyme sucrase will always break down the disaccharide sugar known as sucrose. It's also important to note that here, where you see the name of our enzyme, enzymes tend to always end in ASE. And once again, their name is always derived from the reaction in which it catalyzes. So if lactase catalyzes lactose, it's going to sound similar, and it will always have that ASE ending. Now, just like we said, right, catalysts lower activation energy. That's exactly what enzymes do. So here, once again, we're looking at a diagram that's displaying to us actually cell respiration, which is a energy releasing reaction. So you'll notice our reactants tend to be very high in energy. That's our glucose and oxygen. As a result, activation energy without the enzyme is quite high. But when we add an enzyme to help break down the glucose, you'll notice the activation energy becomes much lower, and so the reaction can happen much faster. Now, how do enzymes know where to go and who to react with? Well, the enzymes provide a site in where the reactants or elements, compounds that are reacting, can be brought together. And so this is known as the enzyme catalyzed reaction. These reactants can also be called substrates. Substrates will bind to a site on the enzyme called the active site. It is the active site and substrate that will have complementary shapes, meaning they will fit together like a puzzle piece. And they are held together by weak interactions. Sometimes we can even call them a lock and key model because their fit is just so precise. So here is what we mean. So here's an example diagram of our enzyme substrate complex. This gray looking piece here, this is our enzyme, which has a very specific shape. Here is its active site, which is where our substrate is going to bind to. Once the substrate binds, it becomes the enzyme substrate complex. It then reacts, and it forms our products. And if you also take note, here we're starting with a polymer substrate, right? This is going to be two monomers bonded together. In the end, they get broken down. So this is actually an example of an enzyme performing or assisting with a hydrolysis reaction. Now, what conditions regulate enzyme activity? Well, because the activity of an enzyme depends on its structure, some conditions can actually change its protein structure and affect its activity. Those two conditions are high hot temperatures and extreme pH. What happens is this weakens the weak interactions or hydrogen bonds that hold our substrate and enzyme together, causing the proteins to unfold, disrupting their active sites, and so our protein is then said to be denatured. Its shape has now changed. And so here's just an example of what enzyme activity might look like in, say, a human. Right? Enzymes produced in human cells generally work best at temperatures close to 37 degrees Celsius. That is our normal temperature of the human body. And you'll notice on our graph here, at this highest point, right, that's the optimal temperature because we're getting the most enzyme activity. As we approach that optimal temperature, we can see the rate of reaction increases. But once we go past that optimal temperature, it gets too hot. Our enzyme loses its shape and it denatures, causes the rate of reaction to decrease. And remember, the same thing will take place when we see extreme changes in pH, which we know is the measurement of acids and bases. All right, everybody, that is it for our video. Please make sure to mark this as done, and don't forget to complete the enzyme exit ticket.